We will now discuss the HIV replication process. Once HIV has entered the body, it migrates into the bloodstream and from there into the lymphatic tissue, where it is able to infect its target cell. This target cell is called a T lymphocyte. You may also sometimes hear it referred to as a CD4 cell. T cells are the cells within the immune system which recognize pathogens and call in other immune cells to fight off disease. This is precisely why HIV is so dangerous. It attacks the key cells the immune system needs to work. The diagram above shows a T cell. We will now work through the various steps which HIV goes through when it infects a T cell. The HIV virus has various protrusions on its surface. These are referred to as docking proteins, and are used by the virus to latch onto and enter the T cell. The T cell has various receptors on its surface. Two of these are commonly used by HIV to enter the cell. These receptors are called CCR5 and CXCR4, and can be seen in our diagram above. The end of the docking protein on the surface of the virus has a shape, which fits into one of the receptors on the membrane of the T cell. Imagine how a key fits into a lock. The docking protein is the key and the T cell receptor is the lock. As such, HIV is capable of opening this lock and entering the T cell. A newer class of medications, called entry inhibitors, act on the T cell receptors to in effect change the shape of the receptor, preventing HIV from entering. Blocking the entry of HIV into the cell in the first place is the most effective way of blocking further replication of the virus. Once the docking proteins of HIV have successfully bound with the T cell's receptor, the docking proteins pull the membrane of the virus and the cell together. This allows HIV to empty its contents into the T cell. These contents consist of the virus's genetic material and the various enzymes it needs to accomplish the replication process. Once inside the cell, the ultimate goal of HIV is to reach the cellular nucleus, so that it may insert its genetic material into that of the cell. Only by doing so can HIV replicate. The reason for this is that, like all viruses, HIV lacks the cellular components needed to replicate on its own. Basically, it is a parasite, and until it enters a living cell is technically no more alive than a rock. Before reaching the cellular nucleus, HIV must change its genetic material into a form that can be understood by the cellular organs of the T cell. It does this using an enzyme called reverse transcriptase. The process of changing this genetic material into an understandable form is not very efficient. Many small errors or mutations occur during this process, causing a great deal of genetic variation in the kinds of virus found, even in a single infected individual. This is also why HIV is so hard to make a vaccine for. Many HIV drugs are designed to block the reverse transcription process. In fact, the first HIV drugs available acted upon reverse transcriptase. However, HIV is very clever. It can quickly find a way around the action of these medications, because of its ability to mutate. That is why we must use a combination of different types of drugs when treating HIV, and why those living with HIV must take their medication at minimum 90% of the time, to prevent viral resistance from developing. If the reverse transcriptase process is successful, then the next step in the replication process is for HIV to integrate its genetic material with the DNA of the cell. This is done within the cellular nucleus, and requires the action of another enzyme, integrase. Integrase manipulates the viral genetic material created during reverse transcription, by clipping the ends of it, allowing a link to be formed with the cellular DNA. Yet another class of drugs, integrase inhibitors, are able to block this process. If the integration process is successful, we have one final option in preventing HIV replication. Once inserted into cellular DNA, HIV can use the cell to produce all the components it needs to make new viruses, which will then go on to infect other T cells. These components leave the cellular nucleus in the form of a long protein strand. This strand is cut up into the various component parts of the virus by the action of a final enzyme, protease. A final class of medication, protease inhibitors, can block this chopping up process. By stopping the chopping up of the protein strand into component parts, what remains is a long strand of genetic material which is harmless. This is because the component parts created by the action of protease assemble to form a new virus, which then leaves the cell, and goes on to infect other T cells within the same person, or can potentially be spread to others. It must be pointed out that this whole replication process occurs thousands of times within a single infected T cell. What ultimately destroys the T cell is not the replication process itself, it is the action of the newly assembled viruses leaving the surface of the cell and creating microscopic breaches in its membrane, which eventually destroys the cell. In the same way that a large collection of relatively minor cuts on human body would eventually result in death, as a result of blood loss,